I'm here to introduce Aleksey Kamyshny, Dr. Aleksey Kamyshny. And I basically know Aleksey from 2009 when I accidentally found his name in the internet, and I wrote him in the States asking him if he would help me with sulfur analysis. And he said yes, and we never ever communicated again till 2012. And then it was a very, very fruitful communication, and I'm indebted to most of my analysis to Aleksey because I was doing experiments and he was doing analysis in his laboratory. Aleksey, as he already told you, did his first, second, and third degree in the Hebrew University of Jerusalem. Then he moved to Bremen, Max Planck Institute in Bremen, for his first postdoctoral research, and the second postdoctoral research was in University of Maryland, College Park, where exactly I was a student and I never met him there. And then we both went to Israel, to different universities, and then started cooperation. Aleksey is a very unique scientist. He's also a very unique human being, but not today about this. But he's a very unique scientist because he's the top of analytical chemistry of sulfur. There is nothing he cannot find or analyze. If you have an issue with sulfur, and that was why I brought him twice, is when we go to Aleksey. Aleksey is now in a Ben-Gurion University of Negev. He's heading his own laboratory, which is biogeochemistry, aquatic biogeochemistry. And this small project is actually in the framework of FAST Foundation. When Sevan turned green the first year, I had this intuition that we are going to see something very interesting, and I knew Aleksey wanted to come to Armenia, and I wrote him, and we were successful, and now he is here for this sampling, and it was a very interesting talk.
Thank you. Should we switch slides? We can see better.
Yeah, well.
Good evening. I'm really glad to have the pleasure to give a presentation today. As Natal already told, we worked together in Israel for quite some time, and for last 5 years I have a pleasure to work with Horen Avitisyan, who was finished master thesis in my laboratory, now finishing PhD. And we, as usually as Jews want everyone to come at least once to Israel, such Horen was telling that I have to come at least once to Armenia. And so we combined this with the scientific part because I always wanted to sample Lake Sivan. I thought that there is not enough, absolutely not enough work done. And what we will see today at the end of my presentation, I will show you that it's even less done than I thought, that there should be a huge research effort should be invested in Lake Sivan. But I will start, my presentation will be from 3 parts. First of all, I will tell you a little bit about my city and my university. Then shortly about history of Earth. I will squeeze 4.5 billion years in maybe 10 or 15 minutes, I will try. And then when we will have this map of time, I will speak about biogeochemical time machines and where we travel, and how all this is connected to Lake Sivan. So my university is located here. This is Beersheba, this is Jerusalem. Here is Tel Aviv, so I'm more or less 1.5 hour drive from the center of the country, and we are located in the desert. We have less than 200 millimeters of rain per year. You can see that here everything is green, but here there is also no green spots. And this university was especially founded 50 years ago for desert studies, because our first Prime Minister, Ben-Gurion, told that, okay, we have 2/3 of the country as a desert and we don't use it, so we should use the desert, and for this we need to research the desert, and the university was built as a desert research university, which actually was for maybe first 30 years, and then it was understood that it cannot be really a good university on the world level if we study only desert, and it started to make more wide perspective and to be real normal university. That's how I was hired there as oceanographer, which was impossible maybe 20 years ago at this university. And that's how Beersheba looked 100 years ago. It was small Turkish army camp with maybe 6 or 10 stone buildings. And this is how it looks now from aerial view. And this is now 200,000 population, and we have a soccer club which is playing in European League, and more or less already normal city. It was mentioned even in the Bible because there was the place where the well of Abraham is situated. And but until the mid-20th century, it was actually a very small army settlement. Now it's developed as the center of the desert, and we have a medical center and so on. This is our Prime Minister Ben-Gurion. When paparazzi caught him on the beach, and he's here 80 years old, and wanted to make photo of him, he told, I want that there will be some educational part, and I want to show the young people that you should make sports and be healthy. So he stood on his head, and there is a photo, and we have even a sculpture of him standing on the head. It's the most popular picture in Israel of our Prime Minister. So he had a sense of humor and also was very, very sportive. This is our university. It actually was established, as I told, exactly 50 years ago. 20 years ago, it was still 5,000 students. Now it's 20,000 students. Of them, 8,000 postgraduate students and 2,600 academic staff in the university. This is the aerial view of the campus, and here is our geology building, actually, where we work. This is the medical center, the sports center, and here is the rest of the university. And so about our department, we are quite small department. We have 12 faculty members, 150 undergraduate students and 40 graduate students, 5 bachelor tracks: general geology, dynamic geology, environmental geology, engineering geology, and paleontology, and 4 master tracks: general geology, environmental geology, engineering geology, petroleum geosciences, which I am actually leading, and I'm participating also in environmental geology, teaching environmental geology track. And here is my group. Here you can see Horen, maybe a couple of years younger. And this is the picture of about a couple of years ago in the campus. And now we'll go to the more scientific part. So here you see the movie poster, The Time Machine. Where would you go? This is our main question. And another very important thing that I will remind you later: be careful what you wish for. So when we will travel in time, and now by Biogeochemical Time Machines, we will answer 2 questions. Where should we go? And actually the second questions are actually if we wish to know something, if we really know something, if we really study the things that we would like to study, or we are missing something. So here we have a geological time scale, and why I don't like this scale, because you see how much is it pressed. We have here 4 billion years and here 500 million years. So if you look on this scale, this part looks much less interesting, and really until this period, until Cambrian, there were no very small number of multicellular organisms. Here is the 4 billion years where Earth was populated mostly by bacteria. So if we look on this scale, more or less at the scale, at the same picture, we see that Hadean, that we know actually nearly nothing except the models about this period, is quite large. Then Archean, and then the huge period, more than half of the Earth's history is Proterozoic, where there were already oxygen in the atmosphere, but still no multicellular life. And this is all the period where multicellular life was on Earth. So this is actually our map where we would like to travel. If we put it on the geochemical and biogeochemical perspective, we can see that 4.54 billion years is the age of Earth. Then we have here maybe 3.5 billion years ago. There are discussions maybe earlier, but definitely not later. We had already first living organisms at 3.5 billion years. I will show you the pictures which are definitely living organisms at this period. We don't know really what was before. There is a large scientific discussion on it. And then at least 3.2 billion years ago, we had already first photosynthetic cells. Most likely that was not oxygen photosynthesis, it was sulfur-based photosynthesis, but it was already photosynthesis. Then around 2.1 billion years ago, we have the first eukaryotic cells, then first multicellular organism, and then Cambrian explosion here. If we look on this history of life and compare it to what happened in the ocean and in the atmosphere, we see that until here, until 2.44 billion years ago, atmosphere was anoxic, and then oxygen concentration rose. And ocean, what was in the ocean is even more interesting, because before there was ocean oxygen in the atmosphere, surface ocean and also deep ocean was iron-rich. Why it was iron-rich? Because actually we can have one of three compounds in the ocean: reduced iron, iron 2, or hydrogen sulfide, or oxygen. We cannot have two of them together. We will see on the next slide why. Meanwhile, just believe me. And we had iron-rich ocean. Then, during Proterozoic, we had ocean which was sulfidic, maybe at the shallow part, oxic at very shallow part, then near the shore it was sulfidic, and the deep parts we had still iron-rich ocean. Why actually was it? Because iron we get from hydrothermal activity, from the seafloor. Oxygen we can get from the atmosphere, and if there was no oxygen in the atmosphere, it wouldn't be a lot of oxygen, even there was photosynthesis in the water column. And then hydrogen sulfide we get from the sulfate reduction, and sulfate comes from oxic weathering of the continent. So if we don't have oxygen, we also don't have sulfate in the sea, and we don't have sulfide, which is a product of its reduction. So iron is there. We will see later what it is about the water column, and why cannot we have these compounds together? Because hydrogen sulfide reacts fast with oxygen. Oxygen oxidizes hydrogen sulfide. Hydrogen sulfide reacts in seconds with iron 2 to precipitate iron sulfide. And oxygen also oxidizes iron 2. So all these compounds can be, to some extent, exist together with manganese, but they cannot be two of them at the same time in the water column. So we can have or iron-rich, or as it's called by some scientific language, ferruginous water column, or oxic water column, or sulfidic water column, which is actually Archean, Proterozoic, and Phanerozoic ocean situation. So let's go quite fast through the history of Earth. We are starting from Hadean iron. It's the first 500 million years of the solar system and Earth's history. The age of the solar system is 4.57 billion years. The age of Earth is 4.54 billion years. Then there was a moon-forming impact. The planet of the size of Moon, or even size of Mars, collided with Earth. And there was some splashed material which then formed the moon. It's widely accepted, not all scientists believe that it's right, but now it's more or less consensus that it's what happened, and more data we have on the composition of moon, more we have confirmation for this hypothesis. Then we have and actually all the Earth was molten with this impact. Then we have the oldest zircons. It's a grains less than 1 cent coin, 4.4 billion years ago. That's the oldest material that we have. But then what we had between 3.95 and 3.8 billion years is what's called late heavy bombardment. We had huge numbers of meteorites which hit the Earth. And there are different estimations. Some scientists told that all the Earth was molten once more. We have a lava ocean. which means that if there was life before 3.95 billion years, it was wiped out and we will more or less never find it. Other scientists told that there were still some places which were not molten, although the temperatures were very high. So for now, we should think that all life, if life formed before 3.8 billion years ago, it was wiped out and we cannot likely found it. That's pictures which more or less depict how moon forming impact looked like and how then Earth looked like during the late heavy bombardment, and then at the end of late heavy bombardment, the moon was very close to the Earth. When there were first oceans, the length of the day was 6 hours, and it looks like the waves were maybe 100-meter high waves because of the moon. Because moon was so close to the Earth. Now we're going to something more close to us, where we already can study Archean ion from no, this is an error. It's from 4 to 2.5 billion years ago. We have Aarchean, Palearchean, Mesoarchean, and Neoarchean. And Earth looked like this. We see no green, no life. Barren rocks, but there was already life in the ocean. There were not a lot of continents, yet there were maybe huge islands or very small continents, and there was an anoxic atmosphere, iron-rich ocean, 100 micromolar of iron 2 there was a concentration. It was very poor in sulfate, from 5 to 200 micromolar. Today, concentration of sulfate in the ocean is 28,000 micromolar. So it was from 2 to maybe 4 orders of magnitude less sulfate than now. Small continents in the vast ocean and the first living cells. Here we can see an ashtray which is carved of a bended iron stone from 3.5 billion years old Barberton supergroup, and we can have these iron-rich formations only in case when we have iron-rich ocean, and all industrial iron deposits are more or less from the end of Archean. We will see it later. And here are the first, maybe here the Strelly Pool in Pilbara Crater in eastern Australia. Maybe this is the first Earth's oldest exposed land surface, not the ocean. Is from 3.5 billion years ago. And here is the oldest trace of life, which more or less all scientists agree that this is some living cell based on the isotopic analysis, shape, and elemental analysis. It's 3.4 to 3.35 billion years ago, most likely 3.4 billion years ago. There are traces which we can say that they look like living cells even 3.7 billion years ago, but on this, scientists still have a discussion. Some agree, some don't agree. The problem is that we just don't have enough material, enough analysis done, enough data. So I think that in future it will be more clear. Now we jump to Proterozoic ocean. It's once more sorry for this. It's from 2.5 to 0.5 billion years ago. We have Paleoarchean, Mesoarchean, and Neoarchean, and then Proterozoic, and then Phanerozoic. That's how Earth looks like in Proterozoic. We can see here bacterial mats, which are called stromatolites, from the Greek word stromatos, which means a mattress, and litos is a stone, so mattress stones, which actually are huge bacterial nets, which can be found only at the conditions of Proterozoic. Today on Earth we have only 2 places where they can survive, one in Bahamas and one in Australia, because we should have a very thin range of hydrographic conditions where they survive. So, 2 atmospheric oxygenation events at the beginning and the end of Proterozoic, anoxic iron deep ocean, intermediate sulfate. We really don't know what was sulfate. So we know that it was maybe as low as in Proterozoic and maybe as high as today. So here are X's, which means that we still don't know. It's under discussion. It's a lot of data which show both directions. Most likely what happened is that there were huge fluctuations in sulfate concentration together with fluctuation with oxygen, and scientists at CLO, when we analyze rocks at similar times but a little bit different, we see large difference. Large continents and first multicellular organisms. Here we can see oxygen concentration, it rose, there was overshoot after the great oxygenation event, and then it was what, between 2 and 1 billion years, which with a little bit grim sense of humor, scientists call Boric billion, because it's more or less nothing happened there. It really was not so boring as we learn now. And here we can see banded iron formations. We can see that there are not a lot of them in the Archean, and then we have a huge peak at Proterozoic, exactly at the place where atmosphere was oxygenated. So all iron which was in the ocean during this time was precipitated as banded iron formation, and we are starting to have sulfide-rich oceans. So we have an abrupt slope around 1.8 billion years ago of banded iron formations. This we won't discuss, it's more connected to the snowball Earth events, but we don't have time to discuss it really now. But more or less here, iron-rich ocean ceased to exist. And then we have the first great oxygenation event. There were 2 oxygenation events at the end of Archean, beginning of Proterozoic, and at the end of Proterozoic, beginning of Cambrian. So the first, which is called great oxygenation event, happened 2.5 to 2.4 billion years before present, and concentration rose from less than 0.005% of present atmospheric level to more or less 10% of present atmospheric level, at least 10,000 times. So, what are the mechanisms? They are photosynthesis, burial of pyrite, burial of organic carbon, and hydrogen escape to space. If we really think that to have oxygen in the atmosphere, we should have photosynthesis. But this is not enough. Photosynthesis was on Earth a long time ago, a long time before the great oxygenation event. Why photosynthesis alone is not enough? Because here we see one of the confirmation that there was a really great oxygenation event at this time. These are sulfur isotopes, and if we are on this line, we have mass-dependent fractionation. If we're out of this line, we have mass-independent fractionation. And we can have mass-independent fractionation only in the atmospheric processes. And only if we have strong UV radiation. So what happened here? Oxygen-rich atmosphere resulted in the beginning of formation of ozone layer. Ozone layer stopped UV radiation. When UV radiation was blocked, there is no more photochemical reactions with sulfur, and if there is no photochemical reactions with sulfur, we have no mass-independent fractionation. So this is one of the best results which show data compilations which show that there was oxygenation of the atmosphere. So if we have photosynthesis reaction, water and carbon dioxide and light energy, we form organic matter and oxygen. And if we go to the respiration reaction, it's totally back reaction of photosynthesis. Organic matter plus oxygen, water, CO2, and energy. So, if there is photosynthesis in the ocean, and then there is some other bacteria which respire organic matter, and we have 2 reactions balanced, we have 0 discharge of oxygen. It's produced in the water column by, let's say, at day by photosynthesis, at night it's consumed by respiration, and although there is a lot of photosynthesis maybe in ocean, there will be still no release of oxygen from the ocean to the atmosphere. So what really we need to get an oxic atmosphere? We need some disbalance. How can we get out of balance? By burying, or we should get from this system water atmosphere some reduced species out. How can we get them out? Or to get hydrogen, which leaves the Earth to the space, because hydrogen molecule is very light and it can leave the Earth and go to space. or we should bury for a very long time reduced species like pyrite or organic carbon into the sediments, and then, due to plate tectonics, even deeper. And in this way, we for a long time, on the scale of hundreds of millions of years, leave more oxidized atmosphere. And so if we do this reaction of photosynthesis, we add sulfate reduction and formation of pyrite, we can get, as a result, formation of pyrite, which is buried in the marine sediments, and net release of oxygen to the atmosphere, which is actually results in the forming of the Earth's atmosphere. Here we can see one of the how it can be shown that there was really oxygen formation and photosynthesis before the great oxygenation event. Here are stromatolites. And here are stromatolites which are grown in the lab, and here is Proterozoic stromatolite, which was formed during the great oxygenation event. And we can see these small bubbles, which are actually oxygen in the stromatolites. We can see the concentration of oxygen. We can have small bubbles of oxygen. And we see very similar bubbles in the ancient stromatolites. That's one of the evidence. We have a lot of lines of evidence. I want just to provide one which is very easy and very fast to catch. This is one of the works which show that there were already photosynthetic production of ocean. That's how look stromatolites from today. In the these are shark-based stromatolites at the Western Australia. Here are stromatolites above the water. Here are stromatolites below the water. And as I told, there are only two places on Earth where we have stromatolites today, in Bahamas and in Australia. And this is how Proterozoic stromatolite looks and preserved. Once more, there are also analysis of not only the shape, but the shape this shape can be formed also by the earthquakes or just by the strain of the sediment, but also composition, chemical and isotopic composition of these layers show that there were photosynthetic organisms here. And now we get to the second oxygenation event, which happened between 0.8 billion years ago and Cambrian. Level rise 10 times from 10% of present atmospheric level to atmospheric level. There are two mechanisms proposed for this event. First, there was, during the formation of Gondwana, we had the super mountains, much higher than Mount Everest today, maybe 10, 12 kilometers high, which were very fast eroded. Then, because of the fast erosion, we have a large flux of essential nutrients, including phosphate, to the ocean, explosion of cyanobacterial population, and more photosynthetic oxygen released to the atmosphere, and more organic matter and pyrite burial. The second theory said that there was evolution of multicellular organisms, quick sinking through the water column, because let's say the whale will sink faster than bacteria, the dead whale, than dead bacteria, smaller oxygen consumption in the water column, and then more organic matter burial. This is a very interesting theory, because usually what we thought that there were release of oxygen, higher oxygen, and then there is enough oxygen for formation of multicellular organisms. Today we think opposite, that there was evolution of multicellular organisms, and there was a positive feedback. Maybe first sponges formed, then this process, all this process started, oxygen rose, more multicellular organisms were formed, so life was not shaped by increase in oxygen, by geologically driven increase of oxygen, but maybe on opposite, the release of oxygen was intimately coupled with evolution of life. So now we get actually to our time machines. How we know all this? How we can study what was in the so deep past. So methods of study of Earth's past include study of the reaction and their isotopic discrimination in laboratory under controlled conditions. We can take and mix all compounds which we think were in the Archean Ocean, then even to add some bacteria, and to see what compounds will precipitate, what will be composition of the water column, what minerals what we get, what will be their isotopic composition. Another way is looking for chemical and isotopic composition of inorganic components of ancient rocks, which called paleoproxies, or organic molecules of life biomarkers, and then we compare that what we have in the ancient rocks, that what we have in the laboratory. If we have coincidence, if we have the same isotopic and elemental composition, then the ocean was like what we had in our flask. What is really, from my point of view, is the wrong side of this approach. Actually, what we make in the chemical flask under controlled conditions should it really correlate with the system which is as complex as the world ocean. So maybe we just mimic something, but we mimic one process, and here hundreds and thousands of processes occur. So actually what we need is a sort of time machine, which will be in between, which we can go and take the modern system, which we think is like the ancient ocean, and study it in details. We can study all the chemical, all microbial processes, and then compare not our flask to the ancient rocks, but modern lake to the ancient rocks. And this approach which I advocate and which I use in my research. So we are looking actually for what we call modern analogs of ancient ocean. Definitely our modern lakes are very different from the Archean ocean just because we have oxygen in the atmosphere. So we should concentrate on their similarities and then study them very thoroughly and think where our approach works and where is the border of our approach, where we should stop and say, no, it's also different. We cannot study everything and not to drive to too serious, too important conclusions even from this approach. So in various modern aquatic systems, some parameters, temperature, microbial communities, and especially chemical composition are similar to those that I believe to have existed in ancient ocean. Research of chemical and microbial processes in such systems should be used to fill the gaps in our knowledge, these gaps between the flask and the ancient ocean. Our paradigm is, which also I don't like and I think it's a wrong paradigm, but we will start from it because it's accepted paradigm. Biogeochemical processes which occur in the water column change chemical and isotopic speciation of elements. Then we have a precipitation, diffusion, and these fingerprints are preserved in the sediments and after diagenesis preserved in the sedimentary rocks with some alteration. So this paradigm says what we have in the ancient rocks, sedimentary rocks, are actually is a result of what happens in the water column of the lake. So now we will go one by one from the ancient past to more modern system with our ancient analogs of the system. The first system which I decided to study to get some background is Yellowstone National Park hydrothermal vents, because there, although there are some living cells and living organisms, they actually, most of the processes are chemical because cell counts are very low. That's only a small number of extremophile organisms which can live there. And isotopic fingerprints are more or less of chemical processes, and we get something as a baseline of abiotic processes, fingers of abiotic processes, which we then can compare to biological processes and see their difference. So what we have here is the magma chamber below the Yellowstone, then magma rises, there is a giant volcanic eruptions of supervolcano, but what we have actually here, it's not on the picture, but we have water circulating, it's going inside the earth, inside the volcanic rocks, and then it's heated and erupted as a geysers or as a hot pools in the Yellowstone. So we go to Yellowstone National Park, and done here it was funded by NASA astrobiology program. Went there and make a research. I will show you results on this pool. It's called Cinder Pool. It's very interesting system. If you can see here these black cinders, That's actually elemental sulfur with maybe 2, 3, 5% of pyrite. That's what is black. The bottom of this pool is of molten sulfur. And volcanic gases, mostly CO2, percolate through the bottom, which is liquid actually of molten sulfur, and bring small cinders, small particles, which are empty inside, and they float because they have CO2 bubble inside. Then at some moment they are filled with water, they sink and are molten once more and rise once more. So this is actually a pool with, it's actually a well, a shape of a well, around 20 meters deep. pH is acidic, 3.5 to 4.1. Temperature is 75 to 79 degrees. It's very close to boiling point because in Yellowstone it's high in the mountains and water boiling temperature is lower. And it's rich in hydrogen sulfide, 25 to 29 micromolar. So there was some inconsistency in the results. Spear et al. Found that most of the microorganisms detected in Yellowstone hot springs, including cinder pool, rely on hydrogen rather on sulfur cycle. On the other hand, if we do microbial ecology study, we see that there is a lot of microorganisms which actually rely on sulfur in their cycling. And so we want to see actually if we have some fingerprints which are of microbial origin in the sulfur cycling or chemical origin. Actually, what we saw in the Yellowstone is that we have a chemical fingerprints, and these fingerprints we use later to compare our other results. It was published 5 years ago. Now we go to Archean, to time machines which we can look through into the Archean Ocean. And these are more rare systems because usually iron-rich lakes with iron-rich deep water are much more rare than the lakes with sulfidic water. Because in this case we should have a very iron-rich rocks around and sulfur-poor rocks, which are not very common on Earth. This situation, it's more or less usually volcanic rocks. Not always, but in many cases there are volcanic rocks. The most popular system is Lake Matanyo in Indonesia, Lake Pavin in France, Lake Nurbishana in Norway, it's just outside of the fence of the Oslo airport, and the lake which we studied together with Horen in China, Lake Sihailong 1. We can see that it's a volcanic crater with very iron-rich rocks and sulfur-poor rocks. So this we studied in the last 5 years, and I will speak more about this lake. It's aerial view of this lake. We can see now very nice that it's a crater. It's a small lake, 0.4 square kilometer, quite deep, 50 meters. It's located here on the border between China and North Korea. Once we even nearly canceled our sampling because North Korean nuclear explosions made here, and we had an earthquake in this place. And luckily we came maybe a month after this explosion and were allowed by the Chinese border patrol to come here. So this is a very, very interesting system. And here are some real results. What do we have in this lake? It's 50 meters deep. We have oxygen in the upper 20 meters. There are 3 seasons, August, May, and October. We can see this peak. We always see this peak of oxygen. That's due to photosynthesis. It's oversaturation. We have saturation level somewhere here, and we can have up to twice the amount of oxygen due to photosynthesis in the deeper water. We can see sulfate concentrations, which are around 50 to 100 micromolar. And you remember that I told you that in Archean concentration was around from 5 to 200 micromolar. So we have really system which is similar to the Archean Ocean. And as you remember, I told that iron in the Archean, in the ocean, was around 100 micromolars. And here we, in the deep waters, have around 40 to 80 micromolars, also the same order of magnitude, and also some manganese. What I want you to show here is an isotope data. And actually, this was also analysis just finished by Horen maybe a week before we came here. This is delta S34 data, and this is delta S33 data. I don't want to go in the details how this signal is formed, but what I want to get to the bottom line, that we have some area in these coordinates, S34 versus S33, which is theoretically can be by microbial sulfate reduction. If we are out of this medusa-like shape, that's not only microbial sulfate reduction. In Yellowstone, we're usually somewhere here, and microbial sulfur disproportionation is somewhere here. What we can see in the Lake Highland 1, that when we are looking into the water column, that's what we get. Very good, nice points inside, most of them except 3 points in the microbial sulfate reduction, theoretical and also practical shape. So everything is fine, and we have fractionation of 20, 30, 40, 50 per mil, which is also perfect match for microbial sulfate reduction. And then we go to the sediments. And what we were told by our paradigm, what we have in the water column, we later have in the sediments, and in 3 billion years we come and analyze it. And you can see what we get in the sediments. It's just about 0. It's increased part of this plot, but actually all our points are here. We have totally different situation. Why do we have totally different situation actually? Why do we have high isotopic fractionation here and low isotopic fractionation here? This is a kinetic isotope fractionation. S32 reacts faster than S34. So we have some difference in the isotopic composition. Let's say if we have two baskets. In one we have apples and pears, and we put apples faster than pears from the first basket to the second, we will have different compositions. But when we move all pears and all apples from one basket to another, we won't see any difference, because all what was in the first, now in the second, we have perfect mass balance. So that's exactly what we have in the sediments. All sulfate is reduced. And because of the full reduction of sulfate in the sediments, we don't have low fractionation, but we don't see it actually in our sediment and later in our sediment rocks. So we see on the example of this lake that what was in the water column stays in the water column. It's not recorded in the sediment. And if we go in 3 billion years here to see these points, we'll say, no, there is no sulfate reduction in this system. Everything is around 0. And what happens is really very different thing. So let's go and see how it works with the analogs of Proterozoic ocean with sulfate-rich lakes. Sulfate-rich lakes are much more often found on Earth. And also we have even the sea which is sulfidic, the Black Sea, and the parts of Baltic Sea are sulfidic. So we have a lot of nice systems with different sulfate concentration to study. And what we studied is the Lake Kinneret or Lake Tiberias or Sea of Galilee in Israel. That's the lake which is famous that Jesus was walking on water in this lake, and that's also work master research of Horen on this lake. We can see that it's seasonally stratified. Actually, it's thermally stratified. In summer, deeper part of the lake is around 14 degrees, and the upper part is plus 30, because it's more warm, the water is more light, there is no strong winds, it's not mixed, there are no storms, and the lake looks like this. There is surface mixed layer, then there is a boundary between two layers which have internal waves on them, and then the deeper layer which called hypolimnial. And we can see here concentrations of oxygen and hydrogen sulfide in this lake as the months of the year. We can see that in winter and early spring all the water column is oxic, and around April, May there starts formation of sulfide, which is continuous until January, February, when temperatures equalize and there are strong storms, so the whole lake is mixed and becomes oxy. There are Horen's results from this lake, and we can see here dissolved oxygen. This is the depth relative to the chemocline. Actually, we show not absolute depth, but depth relative to this layer, because as we can see, it can have internal waves, and if we're here or here we can have the different values. So we actually report it as a function of the chemocline depth, oxygen, no oxygen. Here we can see sulfate concentration. It decreases from half millimolar to around 0.4 millimolar. Once more, very similar which was in the Archean and maybe at the beginning of the Proterozoic ocean. Hydrogen sulfide rises with depth. Here we have different sulfur oxoanions, which is the most interesting data is here. It's elemental sulfur, which is formed by anoxygenic photosynthesis, by photosynthesizing organisms which use not, which produce not oxygen but elemental sulfur and start not with H2O but with H2S. We can see here two different trends, definitely below the chemocline. This is the trend during the dark part of the day, and this is the trend during the day. So what we can see that the ratio of these two slopes, which is around 1 to 12, is the ratio between chemical oxidation of H2S and photosynthetic oxidation of H2S. And when we took samples in the lab, to the lab, and incubated them with and without light, we saw exactly this different 12 times. So, actually phototrophs are accountable for more than 90% of oxidation of hydrogen sulfide in the chemocline. And if we are once more in our little bit different coordinates of S34 versus S33, this is a little bit different presentation from the paper which was accepted a week ago to Biogeochemistry. We can see that most of our results are actually in the microbial sulfate reduction rate. And if we analyze sediments, I still don't have this data presented nicely, but if we go to the sediments, we once more see that this -20 difference is actually wiped out nearly to zero, due more or less to the same reasons. So what we actually can say here, that this paradigm, which was used to study, to use lakes as the biogeochemical time machines, what happens in the water column is then recorded in the sediment, is wrong. And it's wrong due to two reasons. First, there is a lot of things happening in the sediment which can totally wipe out the signal from the water column. The second reason is that we make an assumption that diffusion goes from the water column to the direction of the sediment, but usually in the sediment concentrations there are much faster processes, much more microorganisms, much higher concentrations. So actually in the worst case, water column and the sediment just do not speak one to another. In the best case, what we see in the water column is what happens in the sediment, totally opposite to the modern paradigm. So I guess that the situation here is a little bit not so nice as was thought 5 or 10 years ago, that we can understand from the ancient sedimentary rocks more or less what happened in the sediments of the seas, of the ancient seas and oceans, and not what happened in the water column. It's a little bit disappointing, but once more, we should think exactly what we wish for, what we are looking for. We shouldn't think that what we want to explain, we really explain. We should be much more critical when we're using these time machines, because they are not perfect, and we should try to understand what are the boundaries of our possibility of understanding of ancient systems through the modern systems. As usually with the paleoproxies, we have usually, and with all approaches, the graph of how it looks like this. At the beginning, we think that we can explain everything with a new theory, new method. Then we are more and more pessimistic, and we think that nothing can be explained. And then with time, we are getting to something that we can explain really a little bit. So I think that now maybe we're here, but at the end, I think that we really can explain a lot of things using this approach. We just should be much more critical than we were during the last 10 or 15 years. And now what about Lake Sivan? That we actually, we know from the data from 60s and from the data from the last years that bottom waters of Lake Sivan are anoxic. Because of the eutrophication during the last century, it's anoxic, but it was a concentration of sulfate are also around 100 micromolar, very similar to what was in the Archean Ocean. But presence of hydrogen sulfide in Lake Sivan was never reported. I don't know why it was never reported, because it was not there, because no one looked for it. So it's hard to me to say that there is hydrogen sulfide in Lake Sivan now, and it was not earlier. But what I can say, that we for the first time now detected presence of hydrogen in Lake Sivan. And these are the graphs, what we see at the concentrations of oxygen in Lake Sivan. This is the major Sivan, and this is minor Sivan. And we can see that in the bottom waters of both major Sivan and minor Sivan, we don't have oxygen. This data I can present already because it was measured by in situ sensors. Hydrogen sulfide we should measure in the laboratory, we'll measure it in Israel, but we definitely feel the smell of rotten eggs in the water that we pumped from this depth. So we have hydrogen sulfide there, so I guess that Lake Sivan may be used as a modern analog of Earth's ocean during the Archean Proterozoic transition, during the Great Oxygenation event, the most important and amazing event, I think, in the Earth's history, which changed it drastically. So there are time machines, and one of them is located in Armenia. I think that now, when we made a preliminary research on the very small, this FAST grant as we take as a seed grant, we should think where we can get more funding to continue and to see if really every year there is sulfate in the water column, how it changes seasonally, when it starts, when it finishes, to make maybe samplings every month and to see what happens there. And also to see, because we sampled only 1 point at the major Sivan and 2 points in the minor Sivan, to see how different it is from the point of view of aerial view, if there are only the deepest parts or maybe at the shallow parts near the sediment we also have hydrogen sulfide. And I concentrated during my presentation on the biogeochemical time machines part, but we should understand also which immense environmental impact should have sulfidization of the deep layer, because in these sulfidic layers there can be no multicellular life. There will be no fish, there can be nothing except this anaerobic bacteria, which will produce more and more sulfide. So it can be a runaway situation. As we know, let's say in the Black Sea, the sulfidic layer, due to eutrophication, rose maybe by 50 or 60 meters in the last 30, 50 years. So this process should be definitely monitored, not only from the pure scientific point of view as I discussed, but also from the practical point of view, to see how, let's say, eutrophications on the level of the whole lake, or how fish farms on the local level affect these processes, and how rise in eutrophication, also rise in the temperature due to the global warming, should affect Lake Sivan in the next 1, 10, or 100 years for understanding of environmental management. So you're welcome to ask questions, and if you will have questions later, here is my internet site and my email. You can also, if you want questions later, contact me and discuss these questions with me.
Questions?
So if I understood correctly, so you have here very similar processes occurring at very different times.
Yes, these processes are similar, but the systems really are quite different, because in Archean there was no oxygen. Now it happens because the lake is stratified.
Yeah.
So we should definitely take it into account when we explain our results. But microbial process of sulfate reduction or photosynthesis are, we hope that they are the same as they were 3.5 billion years ago. Definitely there were different microorganisms, but their metabolism was very similar.
The research project that you just mentioned about Lake Sivan, what period do you think is a reasonable 10-year research, 5-year research, to get a good sense of the dynamics?
I would say that, let's say, if we have in Lake Kinneret, it's now 30 years of monitoring. And there is the laboratory on the lake, which have 10 or 11 full-time professor-level scientists who are studying microbiology, chemistry, pesticides, everything. So I think that it can be on the lowest level around 3-4 years old project to get some glance, but at the best case, it should be monitored daily or weekly or bi-weekly or something like this for tens of years. Then we get a really good sense, but it's an investment not on the size of the grant, of one grant, it's on the government level.
Did you look at them, did you look at the monitoring data or monitoring center data? So you could look live?
We talked with them.
We talked with them, and we had some data, and they promised that they will share. That there is open data, which we will definitely use when we will write a paper on our findings. There is some open data, and if there is some not open data, we will speak with them and try maybe to get data which is missing.
They are not checking sulfite for sure.
But they're not checking sulfate. I think they're not checking sulfate and iron and manganese, all the redox.
Sulfate they do.
Sulfate they do, yes.
Sulfate they do.
Iron, manganese.
I think only sulfite because it has to be specific preservation and stuff.
My question relates to this proto-cellulose, because this time machine is going in depth, but all levels you are studying are on the, say, all of them are cellulose. Cellulose, yes?
Cellular.
Cellulose,
Yes.
yeah, cellulose. Do you approach to ones which are not cellular? Because that is a big mystery, how cellulose appears here, and this border nobody no one can overcome. And maybe do you have hope to come to some proto creatures which are not cellular?
Yes. I think that's also connects to Westerhäuser's approach for non-cellular life and this. I think that the answer is unfortunately no, because we are going on the metabolism level. We cannot see if it's cellular or not cellular. What we can see in these areas of sulfur isotopes is metabolism level. So if there will be some metabolism of sulfur with enzymes, but still outside of the cells, I think that what gives these areas which I plotted are enzymes. So we can say when there was not specific, but types of different enzymes that we can say. But we cannot say from this approach if it was enzymes inside the cell or outside the cell, it will give more or less the same result. So I think that unfortunately we cannot do it.
In your answer, you mentioned that there are some studying of non-cellular development and so. You mentioned this researcher. Can you say a few words about him? You said
cellular to multicellular.
Yeah.
Yes, that's actually Don Canfield who is advocating this between single cell and multicellular organisms. That's actually Don Canfield. This theory was actually known in '60s, but now Don Canfield is advocating it strongly. That life actually, not geology, multicellular life, which drove the second increase in oxygen concentration. He had done some very nice works trying to measure what concentrate, because if we think that there was, now it's 20% of oxygen in the atmosphere, and we think that in Proterozoic it was 2 to 4%. And he tried to grow modern sponges and to see what is the lowest concentration of oxygen they can survive. And they survive quite nicely at 1 to 5% of oxygen. And if there were ancient sponges which live in low oxygen ocean, they should live even better with this. So there is no reason to believe that in Proterozoic there was not enough oxygen for multicellular organisms. So the question why they developed only close to that. And maybe one, it's very provocative theory, but immediately after the first great oxygenation event, there was overshoot in oxygen. And then there should be multicellular organisms also then, and then they died off when oxygen go off. And really we have what called Francevillean biota in Franceville in Gabon, where it looks like it's prints on the stone of multicellular organisms which are 2 billion years old, but we don't know really if these are organisms or colonies there is a very discussion, but maybe there was already multicellular life 2 billion years ago, and then it was wiped out by the decrease of oxygen. So it's now quite a hot topic and very much under discussion.
One more question. Thank you very much for a very exciting presentation. So, as far as I understood this, generally analogous or isomorphous processes repeat themselves or are similar to one another under very different geomorphological conditions. Am I right?
Yes, because if let's say we're speaking about iron, in the ancient ocean, the iron was from the hydrothermal activity. Today we have it by or from erosion of rocks around, or the one lake which we studied in southern China, Akha Reservoir. It's just pollution of iron because there are some plants which are polluting it with iron. But for now we are interested actually not in the sources but in the absolute concentration to see what happened when we have when we are looking more or less of a post-mortem of all environmental conditions and going onto the level of concentrations which are now there. But once more, as I told, we should, after we get all these results, we should go back and ask ourselves, okay, but now what are the differences? Where from came iron? What is the oxygen concentration above? And then go back and stop ourselves from making conclusions without thinking about these differences. Because at the beginning, once more, we're at this point, okay, we can explain everything. Now I think we are going down to a more balanced approach when we should think about the constraints and not about what we can get.
Because Lake Sevan and Tiberias, they are in very different geomorphological environments.
Yes, absolutely, but there is also not only this, not only geomorphological, but also hydrological, because there are seiches at Lake Sevan and then hydrological missions, and also it's better to use as time machines lakes which are permanently stratified and not seasonally, because then we have something more similar to the ancient ocean, which were permanently stratified. But the question is actually what we can get out from this data, and we can get a lot, but we should be very, very cautious about our conclusions.
Thank you very much.
It's not a question, it's maybe an addition about Sevan and eutrophication, and that's how the idea came. Is if the government does something to stop the eutrophication, then it would really look like the succession of oxygenated lake body from sulfidi. Basically, you get through the evolution of Earth in a few years, right? That's what I'm thinking.
Once more, yes, it's very nice. I think it's very nice idea, and it sounds very nice and provocative. I think that maybe it's not so right once more, because that the reason that the fact that Lake Sevan is mixed every year is so different from what was in the Archean Ocean that maybe we cannot apply it. We should think, bring all these ideas together, then put them when they write the grant, and go hope that scientists will say, no, that's too much, you're too brave, and then if we get funding, to go there and to check if it really works or not. But I think that yes, we should now really try to start throwing in as much interesting ideas as possible, and then critically evaluate them and think about how we are moving forward with this research, because I see really huge potential.
We should keep in mind that Lake Sevan actually is a very small water basin, right? The catchment basin is relatively small, so the flux and the flow is very slow. So that's also some factor to consider.
Yes, we should think about water residence,
right.
Water residence time, because if you will see,
high, right?
yeah, because if you will see this lake, this is the whole watershed.
Yeah.
So watershed is also extremely small. It's only the crater, because then we have the rim of the crater, and the watershed is maybe only twice size of the whole lake. And yes, I think what we actually lucky with Lake Sevan, that's not a lot of iron-rich rocks around. Definitely we don't have iron-rich waters and we have sulfate-rich waters, but even if there were iron-rich waters, there will be no oxic life in it, fish and so on. But what I think we really have to go around and maybe to sample rocks around for understanding of the fluxes through the rivers and isotopic composition of sulfate in rivers which come in, and to understand where from we get the basic fingerprints from which we are then starting all our microbial processes in the lake. Definitely this should be the part of the research, to study rocks and creeks and we come back and we have samples for multiple sulfur isotopes, which I hope Horen will analyze in the next month or two, and then we will be a bit smarter about what we are looking at.
In your last slide you mentioned about minor cell and major cell. I didn't exactly get the point. What you mean by minor and major cell?
There are two basins in the
This is Pope Savannah.
There are two parts of Sivan, which in literature are usually called major, which is the southern part, which is larger but much more shallow. And Sivan. And I think that what we see here, you see that in the minor Sivan, there is a huge drop in oxygen and then increase. This actually shouldn't be, because if there is oxygen consumption, we should see diffusion profile which looks like this. What is here, I guess, is anoxic water which is coming from the major Sivan, because this is exactly the depth of the seal between them, of the boundary between them, around 30 meters. So I think this is an inflow of anoxic water, of oxygen depleted water at least, from the major Sivan to minor Sivan. So I think it may be extremely interesting to study what happens near this. Yeah, but what happens with sulfur and oxygen and all redox elements near the seal when this anoxic water mix with oxic waters. Maybe we can have, I bet that we can have amazing biogeochemical processes there when anoxic water mixes with oxic water. And this should be a very interesting point to study, but we got this data only now and we had not a lot of time this time, so we can check it, but it should be also checked what happens there.
So pretty much after, what is it, 50 meters, you have no oxygen.
I think it's around 23, if I don't remember exactly, 23 meters. Yes, this is 15. It's around 15 we start. You have to have this drop, and there is maybe 23 or 24. So that's around 5-6 meters of anoxic water in the major Sivan, and there is around. We cannot get. We somehow wanted to sample around 80-90 meter depths, but maybe our GPS coordinates were not precise. We just at the 75 meters we hit the sediment. But it was enough actually, because here we have around 20 meters of anoxic water. It's from around 50 to around 70 meters, and if we will get to the deeper parts, I think it will be 30 meters of maybe. So we should actually also calculate based on bathymetry which percent of volume of Sivan is anoxic now, because although it looks like it's maybe 1/3 of the depth, but it's quite a small deep basin. So in the volume terms, it will be much smaller part of sulfidic water, but we should integrate the depths and to calculate.
So going back to your question about the environmental factors, now you get conditions of nutrient loading. What do you expect to happen?
What do I expect to happen? I expect to happen more primary productivity above, more microorganisms which are formed above, more photosynthesis above this green water layer, and then these microorganisms, when they're dead, they're sinking, and when we have huge flux of organic matter, it actually titrates out all the oxygen, and more eutrophication we will have, larger flux of organic matter down we will have increasing depth of the chemocline and larger sulfidic body. And I think it should be actually on the annual scale. That this year we have larger eutrophication, this year we'll have more sulfide, because lake is mixed and everything is wiped out in winter. So what we start next spring is from zero. And this zero actually is the integration of all primary productivity from spring to October. So I think that it should be actually here on the annual level. Definitely in the Black Sea, it's very different because the resident times is huge and it's not mixed. But here I think that in the year that it will be less microorganisms above, less photosynthesis, we will not see sulfide, we see much less sulfide. So we can study it on annual level to try to integrate all bacterial chlorophyll, all the chlorophyll, algae and bacterial chlorophyll in the upper level, and to compare it to amount of sulfide below, and I think there should be correlation between it every year on the year. But for this we should make multi-annual study and compare year to year to check if it's right. Other reason which can be that sulfide is diffusing, part of sulfide at least is diffusing from the sediment, as I told, and then maybe we will have sulfidic layer for longer times, because then there will be preservation for organic matter in the sediment, and sulfide will go from below. We should model all these processes, but we should have more data, and we should sample sediment definitely for all the same parameters, but it's much more complicated, much more expensive, and needs much more effort to sample sediment than to sample water.
This growing depth of the anoxic, it is a cumulative process, right?
On the annual scale here, yes. I guess it should start accumulate somewhere in summer, and at this time it should be peaking, and then lake mixes, and it will be wiped out. It's accumulating on the scale of months.
But in terms of fauna, it's very critical because they will not survive.
Absolutely, nothing can live, nothing which breathes oxygen, fish, or no multicellular, for all multicellular life, hydrogen sulfide is actually a poison. Hydrogen sulfide is only by different studies 2 to 10 times less toxic than hydrogen cyanide, which Agatha Christie used in all her stories. It's extremely toxic compound. One of my friends who opened the core which was under pressure and got hydrogen sulfide release, he was in coma for 3 days just from one breath of what comes from the sediment core. So this is extremely toxic compound, and definitely there can be some organisms which can survive at very low, multicellular organisms which can survive at low level of sulfide by some detoxification processes, plant roots or something like this. But if we take this without these exceptions, multicellular organisms, all organisms which are breathing oxygen cannot survive, definitely not fish in this area. So we're just wiping out part of the volume of the lake for all multicellular.
I have a question about satellite imagery. I'm just interested, have you ever used satellite imagery or airborne data to combine with what you do?
No, I think that what can be interesting done in this direction, that I think that you cannot actually measure concentrations of sulfide or what happens in these depths, because when we go to these depths, a lake is already not transparent. I think at this depth there will be totally no light, or that all this process, not like in Lake Kinneret, which I showed, it will be more or less here, I don't know, but here it will be definitely chemical, chemotrophic processes. I'm not a big specialist in the remote sensing, but I think that what you can measure really from remote sensing is amount of chlorophyll, how green is the lake, and then from this data you can integrate how much chlorophyll you have in the lake, because even if you are doing sampling, you're doing it in one point, and then you want to integrate for the whole lake. So I think that it can be very interesting to get these basic parameters of how much eutrophication, how much chlorophyll you have in the lakes from the imaging, but not this part which I'm dealing for, but it can be somehow connected with it.
Actually, I've already done chlorophyll measurement through satellite imagery for 2018. I have some time series data during that summer time when we had strong eutrophication.
And this year?
2018, last year.
This year. But I think that if we make a project to understand that all of this should be combined together, you can just write down my email and drop me an email later, and I will have your email, and we can definitely discuss it later in depth if we can have some possibility for collaboration here. Because
she's from Armenian GIS specialist from our department. I don't know if you
was introduced or no, but she's working
No,
in the
no, but-
Acopian Center, so.
Okay, perfect, because I think if we go for some project, we should integrate as much possibilities and as much research as possible to tackle this problem.
And also any kind of, there are many, also not only the lake itself, but also the surrounding the lake, anything that potentially can contribute to the sedimentation, things like that. So.
I mean, the chlorophyll is not the only parameter that we can measure through satellite images. There are a lot of possibilities, like dissolved oxygen and other parameters.
Once more, I'm not specialist in this, so I will be glad to discuss it with you in depth, what we actually can measure, because I don't know what can be measured, but I think it's extremely interesting to discuss it.
We're also just starting a new, well, we will be starting in 2020, new project within the EU Black Sea cross-border cooperation. PONTOS is the project, and we'll be using satellite data to analyze, monitor these variables. So we'll have very strong tools to kind of on demand get analyze satellite data. So I think that would be also very interesting.